Hello everyone, I am K. Devavani, Assistant Professor of Microbiology from Government Degree College for Women, Nalgonda. In this video, I am going to present a topic on humoral response. This is also called as HMI, Humoral Mediated Immunity. The main contents of this talk include what is immune response and what is humoral mediated immune response and what are the phases of humoral mediated immune response and the production of antibodies, what are the factors that are affecting the production of antibodies. Let me first explain what is immune response. Immune response is a specific reaction induced in the host by an antigenic stimulus to an infectious disease. The immune reactions give protection against the invading microorganisms, but not always the immune reactions are beneficial to the host, but they may also lead to the reactions that are injurious to the host. The immune response also includes a state of tolerance. That means it is a specific non-reaction to an antigenic stimulus. When the antigenic stimulus is introduced, the immune reaction does not respond to that antigen. That is called as a immunological tolerance. Immune response is two types. One is humoral mediated immune response and the second one is cell mediated immune response. Humoral mediated immune response is also called as antibody mediated immune response because this immunity is mediated by the antibodies. This is also called as antibody mediated immune response. But whereas the cell mediated immunity is mediated by the cells of the immune system, particularly they are T cells. Lymphocytes are here majorly two types, one is B cells, another one is T cells. The immunity that is mediated by the antibodies produced from the B cells is of humoral mediated immune response or antibody mediated immune response, whereas the immunity that is mediated by the T lymphocytes is a cell mediated immune response. HMI, humoral mediated immune response, it gives protection primarily against the extracellular organisms. Humoral mediated immunity, it gives protection primarily against the extracellular organisms. Also gives protection from the viruses that infect through respiratory and intestinal tracts. HMI has a role in preventing the reoccurrence of viral infections and also participates in the type 1, type 2, type 3 hypersensitivity. As explained, immune response not always is beneficial, it also sometimes it is injurious to the host. That injurious reactions that are observed in some individuals is called hypersensitivity, and there are certain autoimmune disorders. So, HMI has a role in certain autoimmune disorders also. HMI, humoral mediated immunity, It is named because it involves the substances that are present in the body in the form of body fluids, which are named as humors. That is why the immunity is named as a humoral mediated immune response. It has functions in giving protection primarily from the extracellular microorganisms and also gives protection against the viruses that infect through the respiratory and intestinal tracts. It prevents the reoccurrence of the viral infections and it participates in the hypersensitivity reactions of type 1, type 2 and type 3 and also in certain autoimmune diseases. Next is the phases of humoral mediated immune response. It includes the production of antibodies because this is the immunity mediated by the antibodies. So, the antibody production involves this of recognition phase, means the antigen is recognized and it should come in contact with the appropriate immunocompetent B cell. The next phase is activation phase and the B cell gets activated by binding to the antigen. It undergoes steps of clonal proliferation and differentiation and the next one is secretion of antibodies. The antibodies will be distributed in the tissues and body fluids and shows its effects. The major phases of HMI include the recognition phase and the activation phase and the secretion of antibodies. The production of antibodies also brought about by the different types of cells. They are antigen presenting cells and T cells even. In the recognition phase, in the step of recognition phase, the B cells which are having the surface receptors in the form of IgM and IgD, they recognize the antigen and they bind to the antigen and it leads to the activation of B cell. The activated B cell is produced after binding of an antigen to the B cell which is in circulation. As the B cell is activated, it undergoes blast transformation. That means it includes the steps of clonal expansion. Clonal expansion means it produces the clones of it activated copies of B cell. It leads to the generation of earliest antibody that is produced is a IgM and later the isotype switching occurs leads to the production of IgG and here the B cells will be differentiated into the effector B cells and memory B cells. Memory B cells have high affinity to bind to the antigen and effector B cells, they show the reaction of neutralization or killing of the organism. B cells also have a function of antigen processing. What is an antigen processing means? Here antigen is captured by the antigen presenting cells. The antigen presenting cells are macrophages, B cells and dendritic cells. The other way of activation of B cells is the processing of antigens by the antigen presenting cells and their presentation in association with MHC molecules to the T cells. Antibody production involves the recruitment of T cells and that leads to the activation of B cells is is the another way of activating the B cells. The antigens are two types based on the involvement of T cells: T cell dependent antigens and T cell independent antigens. T cell independent antigens are those antigens, they activate the immune system without the involvement of T cells, but whereas T cell dependent antigens, they lead to the production of antibodies and mediate the immune response with the involvement of T cells. For the T cell dependent antigens, first step here is a antigen processing. Antigen processing and presentation. The antigen is first captured by the antigen presenting cells and they are processed and they are expressed on the surface of the antigen presenting cells along with the MHC2. This processed antigen, when presented on the surface of these antigen presenting cells in association with MSC molecules, then that is recognized by the T cells. The antigen has to be presented complexed with MHC2 for the recognition by the TH cells. But whereas for CD8 cells, TH CD4 cells, CD8 cells, the antigen to be presented along with the MSC molecules 1. So, MSC1 is present in all the cells, but MSC2 is present in the only on the antigen presenting cells. Now, the TH cells, after binding to the antigenic peptide along with the MSC2, it makes the T cells to get activated. The activated TH cells or the T cells, they produce cytokines, they release cytokines. The activated TH cell, they release cytokines, thus those include IL-2 and the cytokines for the B cell stimulation. The cytokines that stimulate the B cells are IL-4, IL-5 and IL-6. These cytokines, they act as a B cell activating factors and B cell differentiation factors. So, these cytokines, they activate the B cells to proliferate clonally and to differentiate into the antibody secreting plasma cells. A small proportion of activated B cells, instead of transforming into plasma cells, they will be converted into memory cells, which have a role in the secondary type of response on subsequent contact with the same antigen. There are two types of antigens, the T cell dependent antigens and T cell independent antigens. T cell independent antigens, without the involvement of T cells, the immune response is produced by the antibodies by binding directly to the antigen, and that's get activated. The activated B cell undergoes steps of clonal proliferation and differentiation that leads to the production of effector B cells and the memory cells. The other way of antibody production is by the T cell dependent antigens, which are captured by the antigen processing cells, which present the antigenic peptide along with MHC2 on its surface, which are recognized by the T cells. CD4 T cells, they recognize the antigenic peptides if they are associated with the MHC2, whereas the CD8 T cells, they recognize the antigenic peptides if they are presented in association with the MSC1. Activated Th cell secrete cytokines. Those cytokines include interleukin 2 and the cytokines that stimulate the B cell, which include IL-4, IL-5 and IL-6. These cytokines, they activate the B cells which are combined with the respective antigens to proliferate, to produce more copies and differentiate into antibody secreting cells that are plasma cells and the long lived memory cells. This is about the production of antibodies in the humoral mediated immunity. The antibody production follows phases. Those phases are lag phase, log phase, and this is a steady phase, and this is a decline phase. The antibody production follows different phases. They are lag phase, log phase, steady phase and the decline phase. The lag phase is the phase observed immediately following the antigenic stimulus, during which there is no production of antibody. The detectable antibodies are not observed in the circulation. That phase is called as a lag phase. The second phase is log phase, in which the immune system responds and produces the antibodies to its specific antigen or the epitope that increases the antibody concentration in the serum. So, the titer of the antibody rises is more here in the logarithmic phase. In the steady state, there is an equilibrium between the production of antibodies and the catabolism of antibodies. In the phase of decline, during which the metabolism of the antibodies is more than the production and the titer of antibodies falls gradually, that is called as a decline phase. So, these 4 types of phases are observed in the primary immune response. What is the primary immune response means? The antibody response to an antigen for the first time is called as a primary immune response. In response, these 4 types of phases will be observed. Secondary response means it is the immune response to the same antigen for the second time. Second time exposure to the same antigen, and then the immune response at that time is called as a secondary immune response. The primary immune response differs from the secondary immune response. The differences between the primary immune response and the secondary immune response here is it is the lag period. Lag period is the initial period in which there is no antibody production, generally that is about 4 to 10 days in the primary response, because antigen enters into the host for the first time. The antibody response to the antigen for the first time has a lag period of 4 to 10 days, but for the second time it is only a short duration of 1 to 3 days, because the immune response for the second time is very rapid and fast. And in the primary response, naive B cell is involved. That means the B cells that are produced and that are in circulation which have not come in contact with the antigen earlier are involved. But in the secondary response, the memory B cells are involved. Memory B cells are those cells that are all produced from the B lymphocytes which have already bound to the antigen, undergone the steps of clonal proliferation and differentiation. So, they have the memory of that specific epitope or the antigen. So, those memory B cells are involved in the secondary response for the second time contact with the same antigen. The peak response time is 7 to 10 days. The higher concentration of antibody levels will be seen in 7 to 10 days, but the higher concentration of antibodies will be for the 3 to 5 days only in the secondary response, and in the primary response the earlier antibody is a IgM antibody, but in the secondary response the IgG predominates. IgM has a short half life, so after the IgM IgG predominates, but here in the secondary response IgG predominates. The antibody affinity is low for in the primary response, and for the secondary response the antibody affinity is high. Affinity means here the binding of antigen to the antibody. In the secondary response the antibody binds to the antigen more firmly, that means the dissociation is less compared to the primary response. High affinity binding is due to the mutations that occurs in the DNA that encodes the antigen binding sites. This process is called as a somatic hypermutation. The high affinity binding is due to the mutations that encodes the antigen binding sites. It is called as a somatic hypermutation. Primary response, high dose of antigen favorably with adjuvants is required. What are adjuvants? Adjuvants are the substances that enhance the immunogenicity of an antigen. They are the substances that enhances the production of an antibodies and to an antigen. So, in the primary response adjuvants with the high dose of antigen is required to produce an antibodies, but for the secondary response adjuvants are not required because already it has a memory of it. This is about the comparison between the primary response and the secondary response that is mediated by the antibodies. Now moving on to the factors affecting the antibody production. The first one is the genetic factors. There is a difference in the immune responses to the same antigen shown by the different individuals, and that is determined by the genetic factors. The differences in the immune response are controlled by genes. They are called immune response genes. They are located on the short arm of 6th chromosome. If an individual responds to an antigen, then they are called as responders. If an individual do not respond to an antigen, then they are called as non-responders. So, the differences in the immune response shown by different individuals is determined by some genes. They are called immune response genes, which are located on the short arm of 6th chromosome. The second factor that is influencing the production of antibodies is age. The embryo is immunologically it is immature. During the embryonic life, the lymphoid cells they come in contact with the antigens present in the tissues of the body, and the clones of cells that have specificity towards these self antigens are removed. In the embryonic development, what happens means the developing lymphoid cells they will come in contact with the antigens of the tissues of the body and leads to the production of clones of cells that have the specificity to bind to the self antigens, and those clones of cells will be removed. That is how the immune cells are able to recognize the self and non-self antigens. So, the immune response is against to the non-self antigens. If the immune response occurs to the self antigens, that leads to the certain immunological disorders, particularly that case will be observed in the autoimmune disorders. Immune competent cells are those cells they have the ability to recognize the non-self antigens and mount an immune response. That kind of immune competence is not acquired at birth, but it continues to develop as the infant grows. But the infant has to depend upon the maternal antibodies from 3 to 6 months of his age. The full competence is acquired only by about 5 to 7 years. The full competence, immunocompetence, is gained by an individual by about 5 to 7 years for IgG and 10 to 15 years for IgA. The third factor is nutrition. Nutrition also affects the immune response. Malnutrition affects the immune response adversely. Protein, calorie, malnutrition. It suppresses both humoral mediated immunity and cell mediated immunity. Deficiencies of amino acids such as tryptophan, methionine, glycine, isoleucine, and vitamins such as vitamin A, B, folic acid, they have an effect in decreasing the antibody synthesis. Next factor is the route of an antigen administration. Humoral immune response is better for parenteral administration. Parenteral means by means of an injections, not through oral route. So, the humoral immune response is better for parenteral administration of antigens than the oral and nasal routes. Large particulate antigens are more effective when they are injected into the tissues. For production of IgA, oral and nasal route is suitable. Inhalation of antigens, inhalation of pollen antigens, they induce the antibody synthesis, they are of IgE, and whereas the same antigen, if it is administered and it is given parenteral route, it induces the synthesis of IgG. So, the route of administration and the route of entry of antigen is also important for the generation of antibodies. Next one is the dose of antigen. Antibody production is always dose dependent. Minimum critical dose of antigen is required to stimulate the optimum production of antibodies. If it is the high dose or it is the small dose, it fails to stimulate the immune system, then that is called as immunological paralysis. A very high or very small dose that fails to stimulate the immune system, that is called as immunological paralysis. A single injection of an antigen helps to prime the competent cells to produce particular antibody. The first injection here, it is a priming dose. It primes the immune competent cells to produce a particular antibodies, and the subsequent injections induce the more production of antibodies. They are called as a booster doses. That is the reason for non-living vaccines are given in multiple doses for active immunization. For the live vaccine, single dose is sufficient as the organism multiplies in the body and provides a continuous antigenic stimulus that acts as a priming and booster doses. A single injection of an antigen helps to prime the immune competent cells to produce the particular antibodies. The first injection is called as a priming dose and the subsequent injections are called as booster doses. Booster doses are generally given for non-living vaccines, they are killed vaccines. They are given multiple doses for active immunization, but for live vaccines a single dose is sufficient. Next factor is multiple antigens. Antibody response generally vary when two or more antigens are given simultaneously. The antibody response in such conditions, when they are given in multiple antigens, it may lead to enhanced production of antibodies or it may be diminished. If it is diminished, that is by means of antigenic competition. When two bacterial vaccines such as typhoid and cholera vaccines are given in a mixed form, the antibody response to each is not influenced by the other. But for some cases, there may be a diminished production of antibodies, that may be is of due to antigenic competition. And the next one is adjuvants. As explained, adjuvants are the substances that increases the immunogenicity of an antigen. Adjuvants, they give immunogenicity to non-antigenic substances and they increase the concentration of circulating antibody and also it induce the cellular immunity. Example of substances that are used as an adjuvants are aluminum salts of phosphate and hydroxide, silica, beryllium sulfate, fronts, incomplete adjuvant. Fronts incomplete adjuvant is where the protein antigens are incorporated in the water phase of an water in oil immersion. Here the protein antigens are placed in the aqueous phase, that is water phase of water in oil emulsions. Emulsions are prepared in this emulsion in the water phase, the protein antigens are incorporated and that is used as a French incomplete adjuvant. The potent adjuvant is a French complete adjuvant. French complete adjuvant. It tubercle bacilli is used in the incorporated in the incomplete adjuvant, that makes the French complete adjuvant. It is more potent in increasing the immunogenicity of an antigen. Last one is immunosuppressive agents. Immunosuppressive agents are those that suppress the immune response. The immune response is suppressed in the conditions of surgeries, particularly in transplantation. They include X radiation. The antibody production decreases after 24 hours of receiving radiation. Antigen is given 2 to 3 days before the irradiation, then the antibody response is actually enhanced. Primary immune response is more radiosensitive than the secondary immune response. The second immunosuppressive agent are radiometric drugs. Radiometric drugs are the agents with an action of resembling to that of X-rays. They are alkylating agents, cyclophosphamide. If it is given for 3 days, it completely suppresses the antibody response and this selectively prevents the replication of B cells. Another immunosuppressive agent is corticosteroids. Corticosteroids are anti-inflammatory drugs that diminish the response by both B and T cells. These corticosteroids generally they suppress the delayed type hypersensitivity and inhibit the and suppress the release of histamine. These corticosteroids they also suppress the delayed type hypersensitivity which is a type 4 hypersensitivity which is mediated by the T cells, and the corticosteroids, they also inhibit the release of histamines, and they also change the maturation of activated cells by suppressing the production of antibodies. Immunosuppressive agents are antimetabolites. Antimetabolites include the substances that interfere with the synthesis of DNA, RNA or both, and inhibit the cell division. Cell division and differentiation. They are necessary for HMI and CMI. Antimetabolites are those substances that interfere with the synthesis of DNA, RNA or both, and inhibit the cell division and cell differentiation, which are necessary for HMI and CMI. Examples of such antimetabolites are folic acid, antagonist, analogues of purine, cytosine and uracil. Used antimetabolite is cyclosporine. Mostly used drug is cyclosporine that selectively inhibits the TH cells. The other immunosuppressive agent is Antilymphocyte serum, ALS. It is an anti-serum raised against lymphocytes. That is why this is called as anti-lymphocyte serum. This anti-lymphocyte serum, it acts mainly against the circulating lymphocytes, but not against the lymphocytes in the lymphoid organs. They are also majorly used to prevent graft resections in transplantation. Grafts means they are the tissues or the organs transplanted to another person. Then there will be a rejection. So, to prevent such kind of rejection in the transplantation, these immunosuppressive agents are used to suppress the immune responses. Key points here is what is humoral mediated immune response. Phases of humoral mediated immune response, they are by T cell dependent mechanism and T cell independent mechanism. T cell humoral immune response and the factors that are influencing the antibody production. With this, I am concluding this presentation on humoral mediated immune response. Thank you for watching this video.